DNA replication in prokaryotes and eukaryotes. The process of DNA replication is complex and involves many cellular functions and several verification procedures to ensure fidelity in replication. Replication patterns are somewhat different in prokaryotes and eukaryotes. This process is almost certainly more complex in eukaryotic organisms than prokaryotes. The synthesis of a DNA molecule can be divided into three stages: initiation, elongation and termination. Initiation. In E. coli, replication begins at a single point called the origin of replication. The E. coli replication origin, ORI-C, it consists of 245 base pair, that is highly conserved among bacterial replication origins. There are two series of short repeats, 3 repeats of a 13 base pair sequence and 4 repeats of a 9 base pair sequence. About 30 proteins are involved in the replication of the E. coli chromosome and at least 9 different enzymes participate in the initiation phase of replication. They open the DNA helix at the origin. Now look at the table. The first protein, DNA protein, which is 4, recognizes ORI sequence. It is open duplex at specific sites in origin. DNA B protein, which is 4, unwinds DNA. DNA C protein, it is required for DNA B binding at origin. Then HU, this histone-like proteins, DNA binding protein and stimulates initiation. The next one is primase, that is also called DNA G protein, which synthesizes RNA primers. The next is single stranded DNA binding proteins, that is SSB, which is for binds single stranded DNA. RNA polymerase, which is for facilitates DNA A activity. DNA gyrase, which is also called DNA Topo Isomerase 2, which is for relieves torsional strain generated by DNA unwinding. The next is DAM methylase, which is for methylates 5' GATC sequences at ORI-C. The crucial component in the initiation process is the DNA A protein. A single complex of 4 to 5 DNA A protein molecules binds to the 4 9 base pair repeats in the origin with the help of ATP and the bacterial histone-like protein HU. Then denaturation of DNA in the region of the 13 base pair repeats have occurred, which are rich in AT pairs. The DNA C protein then loads the DNA B protein onto the unwound region. 2 ring shaped hexamers of DNA B, one loaded onto each DNA strand, act as helicases, unwinding the DNA bidirectionally and start a replication by creating 2 potential replication forks. Synthesis occurs at the replication fork, the place at which the DNA helix is unwound and individual strands are replicated. 2 replication forks move outward from the origin until they have copied the whole replication. That portion of the genome that contains an origin and is replicated as a unit. When the replication forks move around the circle, a structure shaped like the Greek letter theta is formed. Finally, since the bacterial chromosome is a single replicon, the fork meet on the other side and 2 separate chromosomes are released. Rapid unwinding can lead to the formation of supercoils or supertweets in the helix, and this will interfere with further unwinding of the double helix. To solve this problem, there is a group of enzymes called DNA Topo Isomerases, which are responsible for removing supercoils in the helix. In eukaryotes, eukaryotic DNA is linear and much longer than prokaryotic DNA. Clearly, many replication forks must copy eukaryotic DNA simultaneously so that the molecule can be duplicated in a relatively short period. And so many replicons are present that there is an origin about every 10 to 100 micrometer along the DNA. Replication forks move outward from these sites and eventually meet forks that have been copying the adjacent DNA stretch. In this manner, a large molecule is copied quickly. Origins of replication, called autonomously replicating sequences, that is ARS, have been identified and best studied in yeast. Initiation of replication in all eukaryotes requires a multi-subunit protein, the origin recognition complex, is called ORC. Two other proteins, CDC6, which is discovered in a screen for genes affecting the cell division cycle, and CDT1, that is CDC10 dependent transcript 1, which bind to ORC and mediate the loading of a heterohexamer of mini chromosome maintenance proteins, that is MCM2 to MCM7. The MCM complex is a ring shaped replicative helicase, which is analogous to the bacterial DNA B helicase. The rate of replication fork movement in eukaryotes, that is approximately 50 nucleotides, that is only 1/20 that observed in E. coli. Like bacteria, eukaryotes have several types of DNA polymerases. RPA, replication protein A, is a eukaryotic single stranded DNA binding protein, equivalent in function to the E. coli SSB protein. The next is elongation. A new strand of DNA is always synthesized in the 5' to 3' direction. If both strands are synthesized continuously while the replication fork moved, one strand would have to undergo 3' to 5' synthesis. This problem was resolved by Raichi Okazaki and colleagues in the 1960s. Okazaki found that one of the new DNA strand is synthesized in short pieces, now called Okazaki fragments. This work ultimately led to the conclusion that one strand is synthesized continuously and other discontinuously. The continuous strand called leading strand, in which 5' to 3' synthesis proceeds in the same direction as replication fork movement. The discontinuous strand or lagging strand is the one in which 5' to 3' synthesis proceeds in the direction opposite to the direction of fork movement. There are 3 DNA polymerases in E. coli. The first enzyme that Kornberg purified is called DNA polymerase 1 or Pol 1. Pol 2 and Pol 3 were identified in E. coli. Pol 2 may repair damaged DNA, although no particular role has been assigned to this enzyme. Pol 3, together with Pol 1, has a role in the DNA replication of E. coli. The complete complex or holo enzyme of Pol 3 contains at least 20 different polypeptide subunits, although the catalytic core consists of only 3 subunits, alpha, epsilon and theta. Let us see the comparison of prokaryotic and eukaryotic DNA polymerases in this table. DNA polymerase 1 activity requires a single unpaired strand to act as template and a primer strand to provide a free hydroxyl group at the 3' end, to which a new nucleotide unit is added. One of the RNA polymerase called a primase synthesizes a short RNA primer, DNA G protein, is usually around 10 nucleotides long, complementary to the DNA. DNA polymerase 3 holo enzyme then synthesizes complementary DNA beginning at the 3' end of the RNA primer. After most of the lagging strand has been synthesized by the formation of Okazaki fragments, DNA polymerase 1 or RNase H removes the RNA primer. Polymerase 1 synthesizes complementary DNA to fill the gap resulting from RNA deletion. Polymerase 3 holoenzyme also may be able to fill in the gap. Finally, the fragments are joined by the enzyme DNA ligase, which forms a phosphodiester bond between the 3' hydroxyl of the growing strand and the 5' phosphate of an Okazaki fragment. Termination. DNA replication stops when the polymerase complex reaches a termination site on the DNA in E. coli. The two replication forks of the circular E. coli chromosome meet at a terminus region containing multiple copies of a 20 base pair sequence called ter for terminus. The ter sequences are arranged on the chromosome to create a sort of trap that a replication fork can enter but cannot leave. The ter sequences function as binding sites for a protein called TUS, that is terminus utilization substances. The TUS-ter complex can arrest a replication fork from only one direction and halts replication. The final few hundred base pairs of DNA between these large protein complexes are then replicated, completing two topologically interlinked circular chromosomes. DNA circles linked in this way are known as catenanes. Separation of catenated circles in E. coli requires Topo Isomerase 4. It is a type 2 Topo Isomerase. The separated chromosomes then segregate into daughter cells at a cell division. In many prokaryotes, replication stops randomly when the fork meet. The terminal phase of replication of other circular chromosomes, including many of the DNA viruses that infect eukaryotic cells, is similar. The termination of replication on linear eukaryotic chromosomes involves the synthesis of special structures called telomerase at the ends of each chromosome. They are generally consisting of many tandem copies of a short oligonucleotide sequence.